This chapter is going to be about DNA and what DNA is and why do we need it. DNA is genetic material of the cell. The DNA consists of four things. There's A's and C's and T's and G's. We call those nucleotides. And they really stand for adenine, cytosine, thymine, and guanine. How we put these combinations of letters together really dictate what we're going to make. It's almost like a recipe. We could put these little codes together in certain ways and make everything from hair to messages that are going to run a neuron. But what did DNA do? DNA, if you remember it, in us, because we're eukaryotes, we have this nucleus, and the nucleus is where we store the most important things in our body, and that's going to be DNA. So DNA has to be protected because if it goes bad, the cell dies. And the DNA is kind of like a blueprint, and what it does is it makes protein. So it's the instructions on how to make protein. It's also the instructions on when to make the protein. So like, for instance, can you imagine right now in all of your cells, you have melatonin in all of your cells, the instructions on how to make it. But can you imagine throughout your whole day, it was making melatonin? You wouldn't get anything done. So it needs to know that your eyes basically send a message to certain cells, and it says, hey, it's getting darker, and when it's getting darker, you start to produce melatonin. So that's why if you sit in a dark room, let's say a dark lecture hall, and you start to get sleepy, it might be because of the material you're listening to, like this one, or it might be because the room is so dark, and your eyes are tricked into thinking it's nighttime. So it's starting to produce melatonin, and melatonin is what's making you fall asleep. Proteins are what you're making, so the instructions of DNA is proteins. It is basically a bunch of different amino acids that you put together, and if you remember back from our biochem chapter, we talked about the big thing is called a protein, but the smallest components of a protein is an amino acid, and we put those together by instructions, when to put which amino acid here and there, and those instructions are DNA. We bring in proteins all the time. So let's say you eat a whole bunch of chicken or beans or anything that has high amount of protein in it. And you're bringing it into your stomach. Hours later, your body is going to remove the protein that you brought in and turn it into amino acids and turn those amino acids into something new. So it could be that they're going to build more hair cells, or they're going to build some blood, or they're going to build hormones. So you're literally turned from a bean to hormones. So when they say you are what you eat, you literally are what you eat. DNA and RNA structures. DNA is deoxyribose nucleic acid, so it's a different type of sugar where it's positioned. And RNA is ribose nucleic acid. Slightly different. It's just how that sugar is placed. It's a message for our cell, and we'll actually see the two in a minute. The polymer, which many of these things coming together, is what we call nucleic acids. But if we were to break those down to its smallest component, it would be nucleotides. And if you remember back when we talked about DNA, we said that it has 4 nucleotides. Those are going to be adenine, thymine, cytosine, and guanine. That's that A and T and C and G. It's made of 3 parts. There is a sugar. There is a nitrogenous base, those A's and T's and C's and G's, and then there's a phosphate. And the sugar and the phosphate are on the outside, so they call that the backbone, and the inside is the most important part, which is the nitrogenous base, the A's and T's and C's and G's. If we look here, what you could see is that the phosphate group, there's a sugar group. Don't worry about all the little how to draw this and all that stuff. And then finally, there's a nitrogenous base. Here we're just looking at thymine, but remember there could be thymine, cytosine, guanine, or adenine. And when it lines up, what it does is it puts the phosphate and the sugar on the outside, and on the inside is going to be the A's and T's and C's and G's. And in DNA, there'll be another strand on this side, but just for this picture, I wanted to show you a basic photo so we didn't get too confused. And as you can see, there are different types of positions these guys can be in. So like thymine and cytosine are called pyrimidines, and adenine and guanine are called purines. Purines have 2 rings, and thymine and cytosine has 1 ring. So we call those pyrimidines and purines. Purines have 2 rings. Watson and Crick are the ones that originally came up with this process. They actually realized that there was something inside the nucleus, and they realized that the DNA was kind of like a rope ladder, but it was spun. And what they realized was that DNA, well, their clues started to put together the impression that DNA was the most important thing in the cell. It is curved and twisted, kind of like what we're seeing here, and it's done this because it allows it to be really compact. It turns out that if we took one of your cells, you'd need to look at that one cell by itself by looking at a microscope. But if we were to take the DNA from one of those cells and just string it from end to end, it would be like a little over 6 feet long. So a cell that you can't even see with your naked eye has about 6 feet of genetic information in there. And it has to, because that cell might only be, let's say, a skin cell, but it has to have the instructions for how to make eye and nose and everything your body does, bone and everything. It might only be using a small subset of those DNA recipes to do whatever it needs to do, but it has to carry everything. So it's about 6 feet long. If I were to take your genetic information and put them end to end to end and line them all up, it would go from here to the sun 4 times round trip. So that's a lot of genetic information compacted into you. If we look at DNA, we'll find out there's something called anti-parallel. And what that means is that, so remember how DNA is double-stranded? DNA pairs up in a certain way. So here you could see guanine. Guanine always pairs up to cytosine. And thymine, the T, always pairs up to adenine. And they always pair up. You'll never see a T hooked up to a C or an A hooked up to a G. It's always going to be A binds with T and C binds with G. So because of that, if you only had one strand of this genetic information, you could put the other strand together. And you'll see that in a second. We'll actually do that in our next video. But because we need to know which end to read, because DNA is only read from one direction. One copy of the genetic material is a redundant copy, and it's designed as a backup. And the other side is the one that we actually use to make proteins and things like that. And it's designed because our DNA is getting attacked quite a bit. So like you go and have UV lights or some chemicals, it'll actually damage your DNA. And what we want to do is protect it. So how we read it is from a 5' to a 3' end. A 5' end always starts with a phosphate. And the 3' end always ends with a sugar. And that's how the body knows that we are looking at the side that we're going to read. The other side we may use or not, but it's really up to what the cell needs to do. Mostly we have one side that we look at the genetic information for, and the other side is redundant. And there's other ways that the cell can tell that this side is the readable side and this side isn't, but I'm just not going to go into it. The other thing I wanted to do with this slide is show you that A and T have 2 of these things called hydrogen bonds that bind them up, and G and C has 3 hydrogen bonds that hold them up. So every time we see a G and C, 3 hydrogen bonds. Every time we see an A and T, 2 hydrogen bonds. And if you remember back from our chemistry chapter, we talked about hydrogen bonds being the weakest bond, and it's easy to open and close them, but they're the weakest bond. And we do this because we have to open DNA and close it quite a bit. We'll talk about that soon.